Hello, everyone. Thank you very much for logging on to our webinar. This is apologies to those that had logged on previously and were not able to access. We had a technical glitch that we're working through to figure out why that webinar ended abruptly. But we're going to be starting again, and thank you very much to Darius and the PDA for helping reset this up. This is going to be recorded, and so you will have access to it at another time with the link that you will be provided by an email. So again, I'd like to start by introducing Darius Pillsbury, who's our speaker today. Darius received his BS in chemical engineering from Tufts. He then moved to a number of small startups where he worked his way through some process development. He was a process development engineer for a few years, and then moved to Eli Lilly, where he was director of process validation and manufacturing technology support. So he was there for quite a while. He then moved to another biotech, Ultragenyx Biopharmaceutical, where he now moved up to director of validation. He then took that experience to Adaptimmune in Philadelphia, and he was director of quality lifecycle management. And most recently, after Adaptimmune, he decided to move to as a consultant to ValSource, where he's now helping a number of different groups with his expertise, which is primarily around quality risk management in cell and gene therapy. And today, he's going to be presenting us with the webinar, the establishment and implementation of the process control strategy, unique challenges for cell therapy products. So with that, I'd like to turn that over to Darius.
There we are. Brian, can you see my screen now?
Yes, I can.
All right, excellent. Thank you so much. So yeah, thank you, Brian, and thanks to Federal Drug Association for inviting me to speak on this topic, and of course, thank you to the PDA Pacific Northwest chapter as well. So my name is Darius Pillsbury. I'm a senior consultant working with ValSource, and predominantly now I am focused on working within the space of cell and gene therapies, or ATMPs, advanced therapy medicinal products, in various aspects: validation, development, manufacturing, process controls, etc. So what I wanted to talk about today is on the establishment and implementation of process control strategy, and really the unique challenges for cell therapy products. So when I was pulling together this presentation, I was doing some reading at the time, and I came across this interesting quote, and it's something that is known as the knowledge doubling curve, and you're going to hear a lot through my presentation. I'm going to mention a lot of key terminology. Of course, you're going to hear a lot about the process life cycle, about process validation, as well as risk management. But a really important piece through all of this is about knowledge and knowledge management. And of course, through time, humans have become better and better with knowledge management and with communication and organization of that type of information and data. And you can see before the 20th century, human knowledge was doubling about every century. Whereas now, and today it's looking like we're doubling about every 13 months, and that's looking to kind of exponentially increase as we continue. So my agenda for today, I'm going to start out by providing a brief introduction to cell therapies within the ATMP or cell and gene therapy space. And then we're going to go through, I'm going to define the process validation life cycle and that approach, and how that can be used and applied to cell therapies. I'll discuss in a little more detail after that a risk-based approach specific to the development of manufacturing processes and identification of better identification of products in order to describe and detail a process control strategy. And kind of throughout this presentation, I'm gonna be adding and interjecting with some examples of various unique challenges that are specific to cell therapies. So that will be kind of dispersed throughout this discussion. Okay, so to start out, ATMPs, or advanced therapy medicinal products. Now I know that's a terminology that is common in the EU and the EMA, and I know FDA as well is adapting this, and there are some nuances between this entire category, or what falls under this umbrella of ATMPs and cell and gene therapy products between the two government bodies. And I'm not here to discuss those nuances, but in general, I just wanted to define kind of the broad categories. So the first of which are vector-based gene therapy medicinal products. These types of products have a healthy gene packaged within some type of delivery system, and what's common now are viral vectors being used. That is then administered to patients, leading to some type of therapeutic, prophylactic, or diagnostic effect. Another category are cell-based ATMPs. These contain cells that have been manipulated in some manner to change their biological characteristics. And they're cells that are not intended to be used for the same essential functions in the body. A key descriptor here is that these cells are somehow significantly modified ex vivo, or outside of the body, and then applied back to the patient. And then another category are tissue engineering products. These contain cells or tissues that have been modified, and they can be used for the purposes of repairing or regenerating or replacing human tissues. Now, for the purpose of my discussion today, I'm going to be focused mainly on the second category here, cell-based ATMPs and cell-based therapies. But a lot of the principles and the approach that I will be discussing can also be applied to the other two types of categories as well. So ATMPs are there are a lot of examples now of ATMPs that are being developed worldwide. You can see from this slide of data that I was able to get through the end of 2019 that you've got more than 987 companies worldwide working on developing what would be considered an ATMP, and 1,066 clinical trials underway. Now we're into May of 2020. Those numbers, of course, have increased. But this is a good example of showing how ATMPs are becoming more and more popular, and that understanding and that knowledge is growing. So as I said, I'm going to focus predominantly on cell-based therapies. So let me give you a little more definition of what those are. So these can be divided amongst either autologous, allogeneic, or xenogenic cells that, as I had noted before, have a significant type of modification to them. Altering of biological characteristics ex vivo, or outside of the body, and then administered to the humans for some type of effect. Autologous cell therapies, these are cells that are derived from the same individual. So the same patient, the same donor, is also the same recipient. You'll hear the term a lot around these of vein-to-vein manufacturing, which means from the same patient is the same product. Allogeneic products are cells that are derived from a donor that are intended for use in another person or other people. So you have different donors and recipients. And then xenogenic cells are derived from an animal source and used in patients. For the purpose of the discussion today, most of my examples are going to be focusing on autologous cell therapies. So let's talk a little bit more about those types of cell therapies. Autologous cell-based therapies, specifically those that have that ex vivo manipulation, these are typically some type of cell, T cell, obtained from a patient through apheresis. There's an ex vivo transduction that's done with a vector that's carrying a gene that is used to equip some type of new genetic information, for instance, like a T cell receptor. Cells are then expanded to target a dose in many occasions. That dose then of product is reinfused back into the same patient. And some advantages here are being able to choose an optimal target for each patient's tumor, and the distinct types of cells that can be engineered, which are important for further this personalizing, this individualized type of medicine. So to date, we do have some examples of ex vivo autologous cell therapy products that have been approved by the FDA. Kymriah was developed and manufactured by Novartis. That drug was approved by the FDA in 2017 for the treatment of patients for B-cell acute lymphoblastic leukemia. There's Yescarta, developed by Kite Pharma, which also was approved in 2017 by the FDA. This was for the treatment of patients with relapsed refractory diffuse large B-cell lymphoma. And a third example I have here is Zynteglo. This was developed by Bluebird Bio and approved in 2019 by the FDA for the treatment of beta thalassemia. So we have some examples of successful development and commercialization of cell-based therapies. Okay, so let's now start to focus our conversation on the product and process lifecycle approach. Here is a typical clinical and regulatory pathway. You can see from here that you have early product development leading to some preclinical studies, which lead into our phase 1 through 3 clinical studies, to the point in which an organization would be ready for licensure or an BLA or MAA application. And if hopefully successful, then into commercialization and post-marketing activities. A key here that I just want to mention is for a lot of these cell therapies and for the indications that these cell therapies are being targeted for, there can be a compressed clinical and regulatory pathway. So those phase 1, 2, and 3, in some cases, may be much faster than for other types of therapies. So overlaying this with the process validation lifecycle, that lifecycle has 3 stages. The first stage, which covers really the early development as well as development of the process through clinical stages, is named the process design stage, which is then followed by process qualification. And in process qualification, there's the product is transferred into a site of manufacturing where, or multiple sites, where that product is then subsequently qualified, and that data then is used in your application for the purposes of commercialization, and following up after that with some monitoring or the continued process verification stage 3 used to ensure that the product remains within a validated state. Now, throughout all of these stages, you have your phase-appropriate quality systems that are overlaid. So it's important to note that there's quality oversight and see where the CGMPs apply, and that those are developed in a phase-appropriate manner. And on the bottom, I certainly want to stress, as I noted before, that really the product process knowledge and the management of that, as well as any risk-based approach and risk management, is built into and should be built into all of these phases of development and manufacturing, until the point of some time where product discontinuation in the future with whatever new novel drugs will be developed. Okay, so just quickly again on as we focus into the process validation lifecycle approach. So process design is stage one, and that's really where you take your culmination of all of your activities around the process and the product and all of that knowledge. That's where that is explored, and ultimately the establishment of a control strategy is put into place. Process qualification, as I mentioned before, it's split into two stages, 2A being qualification of the GMP manufacturing systems for that specific drug, and then 2B being your process performance qualification lots, or what some regulatory agencies and in the past has been referred to as process validation runs or campaign. And the third piece being that monitoring of the validated state in stage 3. So what we're focusing on here today is stage 1 and process design, and that key output from this stage, which is the process control strategy. So some of the benefits for using this risk-based approach and for developing a process control strategy are defined as certainly organizing important information for the purposes of both internal discussions. There's many different business units within an organization that are involved with control strategy development. So organize that information and having that communicated amongst these different groups is very important, as well as for external, especially regulatory communications. Focusing your process development, your process characterization studies, and any type of robustness optimization studies. Control strategy is important for understanding which process steps and systems are critical. And really then that helps to define the scope of any subsequent qualification activities that are needed in subsequent stage 2. Understanding of regulatory requirements specific to that type of product and the process. Supporting manufacturing tech transfers, for instance, to CMOs, contractor manufacturing organizations, as well as within an internal system, and supporting process changes. There are many of those within for these ATMP products and cell therapy products through early development as you continue to improve this process. And an important part as well being the clear understanding of raw materials that have an impact on product quality. So all these are important, and these are benefits for this process control strategy based on a risk approach. This quote I also came across recently. So when we're talking about some of the unique challenges for cell therapies, and specific to these within this whole life cycle approach and within that clinical regulatory pathway, Peter Marks, who's director of CBER at FDA, had said, that in manufacturing, there's a need to focus on producing quality products by design in scalable processes, so that if these early clinical trials are promising, they can advance development rapidly. And so that's a key for these products, and that's something that you're going to hear about a lot as I continue this discussion, is how can CMC meet the goals for this compressed and rapid pathway. So here are some examples of some of the unique challenges that I've seen with many companies. As mentioned, that rapid clinical timeline, the FDA has their RMAT designation to work with companies for rapid clinical development, and there's PRIME within the EU as well. Mechanism of action for a lot of these cell therapy products that may not be well understood when you're embarking upon early clinical studies. So that, of course, a lot has to be explored. Product variability. These products have some unique sources of variability that can impact final product quality. Namely, and as I'm talking today about autologous cell therapies, namely the starting materials that are used. Stability of these products, short shelf lives for some, especially for those that are not successfully being able to be cryopreserved. Scale out as opposed to being scaled up. With individualized therapies, more and more development runs will be needed since there's one patient is one manufacturing lot. So this concept of scaling out the process as opposed to scaling up to larger and larger volumes for multiple patients, as is done with other types of products, like for instance biologics or monoclonal antibodies. Product tracking also being extremely important. One patient, one batch, chain of identity is critical. And understanding of regulatory requirements. These requirements are being developed and implemented as we speak. And earlier this year, FDA put out multiple guidances that are specific to cell and gene therapies. Okay, continuing on key challenges and considerations for cell therapies. And as I mentioned previously in a little more detail here, rapid clinical development and multiple process changes drive the requirement for enhanced process and product understanding. There's multiple functional teams that are required, and I think this collaboration is very important for all types of medicines that are being developed, but especially for cell therapy products, where you've got close communication between your process and analytical development, manufacturing, quality assurance, your regulatory research, as well as with communication with clinical and preclinical. And overall, for your process and product understanding, control strategy development being so important, as well as comparability when we talk about things like assessing process and product changes. So when looking through this life cycle, I wanted to show this slide so that you can see, specific to cell therapies, that when you have your early research and your development, an important next step in that is product and process understanding. And that then goes into your specific process development to better define your manufacturing process and control product quality on a consistent basis. So then that transferring into GMP manufacturing and the data that's generated there being so critical to feedback and have optimization of your development, as well as any of your correlative analysis of patient samples. That too is so important to helping to better understand your quality attributes of your product, which we'll get into more detail about, as well as any type of control of the process and monitoring that's required. So, those types of desired characteristics of the product, where that might be phenotypic characteristics or some type of functional characteristic. And just linking those characteristics to the process parameters and enabling a robust control strategy. This is key to this whole understanding. So let's go through and talk in more detail about overall what should be a part of your control strategy. The first thing you really want to focus on is, what is the product that I am trying to manufacture? That's typically done through something that's called a target product profile. And what that is a strategic development tool that provides a statement of the overall intent of the drug development program. And it gives information about the drug at a particular time in development. That's from an FDA draft guidance. The key here is trying to develop this target product profile as early as possible, and using that to then facilitate a common vision across all of those disciplines that I mentioned in the previous slide, so that you can have this guide to your development, as well as conduct and the types of analyses that are needed within clinical trials. And this ultimately, focuses on strategic product label claims and maximizing the efficiency of that drug development program, which is so important to meeting these quick timelines. So the TPP should be developed at the initiation of product development as early as possible. And really this is focusing on beginning this process with the goal in mind. And here I've got my target, and hopefully that arrow hits that bullseye. So what does a TPP look like for a cell therapy type of product? Well, what I have in this table here are different types of product aspects that you might see in a TPP, and the general considerations that you would want to consider for the purposes of defining for whatever type of cell therapy. Now, of course, there's many within cell therapy. There's many different types, but I think this helps with at least getting asking that initial question and having those involved in development be able to think about these things and be able to set these goals, as we talked about in the target product profile. So I'm not going to go through all of these, but you can get a good understanding here of the types of things that should go into defining that final product goal. Things like defining the therapeutic indication, what's your target patient population, population size, what's going to go into that clinical trial design, product classification, for these types of regenerative therapies, whether you're going to have an autologous or an allogeneic product, and in some, like for instance in EU, considering potential GMO regulatory implications, cell source, things about dose, things about your mechanism or mode of action, how this is going to be administered, what's the strategy for determining dosage and regimen, and even things like looking at market scale and demographics. Now, once a good target product profile has been put into place, we really need to start focusing on what are the quality aspects of that TPP. And what the QTPP is, thus, is it's a prospective summary of the quality characteristics of the product. This is what we ideally want to achieve to ensure desired product quality and consistent product quality. And here's where we take into account and really start focusing on safety from a patient perspective and efficacy. Tactical implementation of the strategic vision should be outlined in the TPP, right? But that's modified then through the product life cycle as knowledge of the drug increases. And the QTPP is part of that. This should be set at initially at the beginning of development, but it will be updated. It is a life cycle document, and thus will be updated as you continue to go through your stage 1 and your process design part of your process validation life cycle. This quote from ICH, I do like, protection of the patient by managing the risk to quality should be considered of prime importance, and that's an important piece of the QTPP. So what do the quality aspects of the target product profile look like? Once again, here's another table that gives some typical types of aspects that should be considered for a cell therapy product, as well as some of the corresponding general considerations when trying to define these for your QTPP. Dosage form, your volume per dose is key. Is this a liquid suspension? What are the types of cells that are going to go in here? How is that going to be delivered to the patient? Stability and storage conditions, of course, being critical to quality aspects. Describing your product storage conditions and having a good understanding of that. We know many cell therapy products, if they can be, if cell viability can be maintained, can be cryopreserved. Container closure, what is your primary product container look like, and how will that be sealed? And really atturance of most of these cell therapy products are going to be sterile as they're infused. Product attributes, so really now starting to break down in general, what is the safety, what is identity, my dose and content, my purity and impurity, starting to think about these types of concepts. And I'm going to go into a little more detail about these as I will be discussing the critical quality attributes for products. But this is kind of the outline for what a QTPP should look like for a cell therapy product. So as I said, CQA. if you're not aware of or haven't worked with CQAs before, defining those, these are a physical, chemical, biological, or microbiological property or a characteristic that should be within an appropriate limit of range or distribution to ensure the desired product quality. These need to be controlled in order to have and ensure consistent desired product quality from batch to batch. And the general categories or subcategories within CQAs, and these attributes that need to be identified for cell therapies, are similar to other types of medicinal products as well. Things like identity, your potency, activity, purity and impurities, your dose or your strength of your product, and microbiological or other types of attributes that could potentially be need to be controlled, especially for, as I mentioned, a sterile product. So now I'm going to go through and describe a risk-based approach for use in determining CQAs. A lot of what I'm going to describe in here is consistent with what PDA has put into Technical Report 81. So a lot of details that I'm describing here. There's even more description within that technical report if you're interested in finding out some more about that. So when we first think about product attributes and we start generating a list based on our target product profile and our QTPP, as I mentioned before, we want to assess the potential impact of those attributes on patient safety, or things like immunogenicity, as well as on efficacy and potency of that product. So an initial way of doing this is a pretty simple risk ranking of low, medium, and high. So assessing the severity of harm and impact to each of to the patient based on each type of attribute. So when I say, what is an example of when we're saying low, medium, high, for instance, for patient safety? Well, low would be there's no indication that based on the knowledge that we currently have for this product, that there would be any patient harm based on this type of attribute. Whereas medium, there may be some adverse event that can be managed by some type of clinical attribute. So there is some risk there. Whereas high, we know that that attribute has a life-threatening or an irreversible adverse event. Same thing applied, low, medium, high for efficacy. And as you can see here, we're talking about the impact on the patient, whether it's marginal or a small potential for impact or significant impact or potential. And really, a lot of this can come from clinical studies that have been ongoing, or just an understanding of this class and these types of products. Important, though, for cell therapies is also trying to assess an uncertainty score. And here we're determining the current level of product knowledge that was used in the assigning of the criticality or that severity score that I just previously talked about. And for a lot of these products being novel and not quite understanding some of the clinical aspects of this, uncertainty is something that needs to be understood so that you can focus your studies and focus your control strategy development thereafter to try and answer the question of, if something is high uncertainty where I have limited scientific understanding, well, how can I develop some studies and how can I get some in-house data, some clinical experience in order to lower that uncertainty score so I have a better understanding of my severity impact score. So using both of these scores, I'm going to apply it to a matrix where, so your impact or severity and uncertainty, they're used to classify criticality, right? And you initially come up with really three different categories for CQA or for a product attribute, which is a non-CQA, a potential CQA, and a CQA. And based on that, these then can be used in, as I mentioned before, trying to design your studies so that you can, if for instance something is a potential CQA, you can focus on that potential CQA and be able to develop the types of studies so that you can hopefully finally be able to categorize and attribute as either a CQA or non-CQA. So just a couple examples here of what I'm talking about. Something like sterility, that we talk about for cell therapies, and sterility assurance for those. We know the impact of sterility, it's very high. These products need to be sterile, and that uncertainty is low. We understand this very well. That would be identified initially, of course, as a CQA. Whereas something like a process-related impurity, it's an attribute of the product. But at the time in which we're assessing the initial assessment of the CQAs, we may not know that much about it. So that impact score may be somewhere medium, and the uncertainty score may be someplace medium. And we know that this attribute needs to be focused on as we go through the process design stage 1. So what are some of the unique challenges now that we understand that risk-based approach for identifying CQAs? I'm always going to be stressing that compressed development timeline. It's really going to put a lot of pressure on your CMC unit to be able to identify CQAs. Once again, mechanism of action, and I mentioned that before. A limited product history. These are novel drugs, so there might be, you're not necessarily going to be able to pull from some type of a product history. Process transfer coming from academia. This certainly, of course, has impact as well on your development of your process, specific to the CQA, and understanding how that would impact some of the quality attributes. Development of analytical tools. It's important to be able to monitor some of these CQAs as part of your control strategy. So this would help with focusing your development of analytical methods and trying to determine specific analytical methodologies, and some of these are new and unique. Data management and integrity. I've mentioned before that it's very important that data and product knowledge are well organized and communicated, and so the more that we can do early on to strengthen data management, getting out of just this management of hundreds and hundreds of Excel files, and being able to have some better assurance and integrity of data, then that will be a benefit for your development program. And as I mentioned, the communication of that data amongst many different business units, it's really important. So some examples here then of attributes that should be evaluated for determining what are the CQAs of a product. And description here that I've given that's specific to cell therapies. Dose and strength, a lot of these are for products so far have been based on kind of the number of transduced target cells, or some minimum percentage, or maybe based on patient weight. Identity, I had mentioned previously before about chain of identity being important. So that's key. That's a key attribute to make sure that the patient is receiving the correct product. Appearance as well, especially when we're talking about things like the potential for visible foreign particulates. Potency, developing that cell product-based potency assay and understanding functional activity. Cell viability being very important here. These products need to be viable in order to be active in vivo. Vector copy number, which has a potential for oncogenesis. Replication competent retrovirus, with a theoretical risk still remaining for patient safety due to replication of retroviruses that may be used for the product. I mentioned before product and process related impurities. There are a lot of examples here of these, as well as any microbiological attributes. Okay, I mentioned before the vein-to-vein process, so, and I had said I was going to get into a little more detail about that, so here that is. So I've got a process map for kind of a high-level manufacturing for an autologous ex vivo genetically modified cell therapy. So That apheresis starts with that individual patient collected typically at a clinic or the hospital. It's then, those cells then are shipped to a manufacturing site where there might be some type of wash of the cells and a cryopreservation if possible. Then there's a step where a certain type of cell will be selected, or T cell perhaps, selection and stimulation. And then the gene delivery by viral vector transduction. And so this would come from where the production process and manufacturing of that specific viral vector used for that transduction. There then typically is some type of cell expansion, once again, to try and meet a certain dose that would be established through clinical trials. Cell isolation and formulation. This is kind of a type of, if you will, a purification or recovery step. And then formulation for the purposes of stability of those cells. For instance, maybe for drug product cryopreservation. And then the QC release testing that's associated with that, so that hopefully if meeting all of those quality related tests, you can ship back to the clinic and hospital and product then administered back to that patient. So vein to vein, one patient equaling one manufacturing batch. So as we look through that manufacturing process and are developing that control strategy for each of those types of unit operations that I just mentioned, a good thing to do is to start with taking that process map and listing your process variables for each of those unit operations. And these typically can be characterized as either input variables, which are operational parameters that can be controlled or somehow modified perhaps directly. And these may be, these are identified as process parameters, as well as output variables. These are dependent variables that are a result of the process. And these are identified as product quality attributes or process performance attributes. And there's some differences that I've seen with different organizations for how these are identified, but in general, these are your inputs and outputs from those unit operations. So now the idea is, once we've got a good understanding of our CQAs and have listed out those process parameters, we want to define the linkage between the process parameter and the CQA. So we know what we do or do not need to control, what is or is not critical. And the question here to ask is, how does that variability of that process parameter, that input, when operated within a certain range, how does that impact the potential variability for that CQA output? And once again, a risk-based scoring can be done for this, where you can look at low or a negligible potential. That parameter has no impact to the CQA. A moderate or perhaps an unknown potential would be kind of in a middle score, where a large change or maybe a small change in combination with another factor could have a significant impact on one or more CQAs. And a high impact. Here, a small to moderate change in the process parameter would result in a significant impact to one or more CQAs. So for initial type of scoring, I would stick to this kind of qualitative approach, whereas when revisiting this in the life cycle and the approach, you could perhaps drill into some categories and maybe use a qualitative system. But for initially, when we're first starting out our process development, I think it's important to look at this qualitative approach. So here's an example that I'm providing of how to take this next step and how to apply that scoring from the table I just showed. This example is one that I created. So this is kind of for a product that does not exist, but is somewhat similar to cell therapies, of course. But there's each cell therapy product is going to have its unique unique aspects, and so an assessment should always be done based specifically on your unique cell therapy product. But some of the types of parameters that are looked at for a unit operation like cryopreservation of cell products are the cell concentration, cryoprotectant concentration that's used, exposure to that cryoprotectant, and the rate at which then the cells are frozen in that cryopreservation formula. So going through and looking at each of the CQAs, and I've got 6 examples here across the top of different types of CQAs for this product example. And this initial scoring, once again, low, medium, high. And coming out of that criticality assessment, then you'll see the classification as either whether something is believed that parameter is believed to be either a potential CPP, a CPP, or a non-CPP. And in this example here, I'm looking at all 4 of these and thinking that there's some potential that these could be a CPP based on the impact to each of these CQAs. And then the idea here is, so this being your initial assessment, is to go in and develop your further process development studies so that you can then be able to better determine from your actual data from your laboratories the better understanding of the linkage between the parameter and the attribute, so you can also then be able to determine a proven acceptable range. And so you might come back after a whole series of process development studies and revisit this assessment and look at it and say, okay, now that I've got that empirical data, I have a better understanding of this relationship, and I can say yes or no, this is a CPP or this is not a CPP. And that's an important part of the establishment of the control strategy. So kind of as you're assessing criticality of your parameters, you should also be looking at how well you can control those parameters. And this also is an important part of your development. So this is a process capability assessment. It's used in the evaluation of the ability to control and detect out-of-range process parameters and determine how well that parameter ultimately is controlled. And really here, I would lean towards a quantitative scoring system, because you really want to be able to risk rank these, your ability to your capability for each of these process parameters. So once again, taking those parameters as, for my example there with cryopreservation, taking those parameters and then further using types of empirical data, which may come from your development studies, it may come from your clinical manufacturing, as well as experience of your subject matter experts, and trying to identify an occurrence or likelihood that you'll exceed the range that you've established, and your ability to detect. Do I have the ability to detect that that parameter immediately within the process, or at batch release, or do I not have that ability to detect? It's impossible. And then using the severity score that we discussed previously, as well as these occurrence and detection, you can identify an RPN number and risk ranking. And this can help with your further development studies, your further studies to better control those process parameters. Okay, so I spoke to some unique challenges as we've been discussing this whole control strategy development piece, but let me just go through again, some of the important nuances that we see for cell therapy products. Yes, compressed timeline. Yes, we have to get things done quickly. Qualifying a representative model is important. A lot of times, because we're at such a small scale for cell therapy products, you might be able to have a lab model that's representative of your manufacturing scale. Using the use of healthy donor cells versus patient cells. Of course, you would want to have an understanding of if you are going to use healthy donor cells, which are more readily available, of course, and patient cells can difficult and cannot be used for the purposes of a lot of these development type of studies and a lot of that product knowledge information. what are the differences between if I'm using those donor cells versus patient cells when I do my, for instance, process characterization studies? The quality of the vector that you're using and limited quantity of that vector. That vector can be very expensive to manufacture. So when you're using that in your manufacturing, in your development studies, you need to do a very good job of designing your experimental studies. And it's not something that I'm going to get into today, design for process characterization studies. I could definitely spend an entire hour on that alone. But there are other resources for exploring those DOE studies. Once again, development of analytical methods is important, and novel analytical methods. Your raw materials and components that you're using and ensuring the quality of those. Important to use those high quality materials and components in your development studies, so it'll be reflective of what you will see, hopefully, in your manufacturing. And then once again, data management and integrity and the communication and organization of that. So hopefully you can get to the point where you have a mature, and what I like to say is a robust process control strategy to then move into your next stages, stage 2 of the process validation lifecycle. Okay, so now that we have identified what's needed from a product knowledge standpoint, your TPP, your QTPP, your critical quality attributes, and developed a process control strategy, so where we have an understanding of your process parameters and controls for that. This information, this knowledge can then be expressed and transferred into other areas of control for what I like to call an overall control strategy. So taking that product and process knowledge that's going to go into the process control strategy and implementing that throughout the rest of your overall control. Things like control of analytical methods and developing an analytical method control strategy. Your raw materials and a raw material control strategy. The facility and equipment, facility design, equipment design, and the subsequent qualification of those for use in manufacturing. Contamination controls. And your transport and shipping, also a very important part, a very important to assure control of that. So I'm going to give some high-level examples of each of these types of control strategies. So let's start with raw materials. Risk-based approach and risk assessment is key in the development of a robust control and testing strategy for raw materials that are used in the process. This flow diagram I took out of PDA TR81 for control strategy of cell therapies. But you can see just how many areas in which you need to focus and use a risk-based approach to understand the control of these raw materials. And I'm talking about processing reagents and excipients. Starting materials, we talked about vector before, as well as starting materials, that patient apheresis, and other types of consumables. I would point your attention to the lower left box, which says the assessment of criticality based on CQAs. An approach that I just described for process parameters, a very similar one, can be also used for the assessment of the criticality of raw materials for the purposes of control strategy development. So looking at raw materials and the criticality to CQAs. From a contamination control strategy standpoint, there's many different sources that potentially can go into microbial contamination, as well as other types of contaminations of your product. It's important to use quality risk management and knowledge management for these. A tool that I really have found and I utilize is called a HACCP, hazards analysis and critical control points. And you can see here the different areas like utilities, equipment, materials, process, facility, and personnel. All of those need to be well controlled in order to assure product quality. And some unique challenges for cell therapy products include, I think I mentioned before, but These cell-based products, there's no terminal sterilization of these, and these are sterile products. So the manipulations of the product throughout the entirety of the process are aseptic. There's a lack of dedicated viral inactivation steps and reduction steps. So you've added that vector, and downstream of that, there's no way of removing anything that could be adventitious or any adventitious viral agents. Product segregation, going back to your chain of identity. You've got multiple products, patient lots, and viral vectors concurrently being manufactured. How do you control all of that? One lot per one patient, going back to our individualized and autologous therapies. Risk of a contaminated batch is such higher severity. You don't want to lose that batch because that one batch is specific for that one patient. Analytical methods control strategy. The life cycle approach also applies here, and a risk-based approach is very important. Once you've got a good understanding of those CQAs and your outputs of your process, you need those analytical methods so that you can monitor these things. So cell products are complex. There's a lot of variability involved. Stability can be an issue. A fast turnaround time is typically needed for release testing, in order to meet individual patient needs. There's limited material. Product in your primary container. You may not have a lot for the purposes of analysis and retaining and testing that's needed. And also for product comparability, it's good to understand, you need to understand those CQAs and have good development of analytical tools to make any of those process changes. And here's an example of some of the CQAs again, and corresponding analytical tests that could be developed. Facilities, utilities, and equipment, a strategy being developed for the qualification of all of these, defining specifications like user specifications that then can be used for the qualification. This is all should be taken a risk-based approach, and really this needs to be dependent on those CQAs, like for example, cell viability being a CQA. You may have a CPP associated with it, for instance, in a bioreactor, like dissolved oxygen. Okay, well, when I qualify that type of system, whether it be, and I just threw some examples in here, I am not promoting any type of equipment, whatever's developed for your product, it's going to have to get qualified. So there might be things from a critical aspect and a qualification standpoint, like agitation speeds in a stir tank bioreactor and air and O2 flow rate. If you're in a rocking bioreactor, how many rocks per minute you're doing, and what's your overlay gas and the content of that. All of this is important, and that equipment strategy for qualification needs to be based on your control strategy. And then last in these categories that I wanted to discuss, unique challenges for cell therapy products around product and transport. Risk assessment is critical for this as well. You need to develop a robust shipping control strategy. For vein to vein, you've got to cover the entirety of it. At the beginning, when you have the apheresis from the product and it's shipped to the manufacturing site, you need to ensure that shipping is robust and that shipping is qualified, as well as on the back end when that drug product then is manufactured and going back to the clinic. If it's cryopreserved, you're going to have some unique considerations for the shipping of that as well. Maintaining product quality throughout, asking that question of what CQAs are potentially impacted. The development of technologies here, I think there's some areas of improvement around control of physical shipping conditions, as well as product tracking, and really getting to know and better control your product. And I point to a risk assessment that I had worked on a while back, and the amount of just experience amount of information and data that went into manufacturing of a product. And when we actually were ready to ship that product out the door, the company for shipping had shown up in a Toyota, I think it was a Toyota Corolla. That's not a picture of it, but very similar to this, and threw it in the back seat. So you could see that, this vein-to-vein piece and all the shipping, the control strategy is so important, getting to know every aspect. Okay, quick summary here. The risk-based approach being applied to the establishment of process control strategy as part of the overall life cycle approach is a good way to focus on what is critical and what is important to your product and controlling that. The approach has many benefits, and I'm reiterating a slide from before. Organizing rapidly increasing product and process knowledge through your process design stage, a focus on critical elements of product and process, identifying what you do not know, the uncertainty piece to this, and then formulating a plan on how to address that. Communication of any new learnings or knowledge across multiple disciplines and externally as needed. Defining roles and responsibilities of the business units, and just readying your organization ultimately for that goal of validation and successful commercialization. So I want to say thank you for your attention on this. And I wanted to point out that the 2020 PDA ATMP products month webinar series is going to be held in June of this year. So every Tuesday and Thursday of June at 11 a.m. there's going to be a different session, and of course this is going to be held virtually. And so it's a series of 9 webinars featuring industry and regulatory experts, where each webinar includes ample opportunities for live Q&A with those presenters. And you can go to the PDA website to learn more about this. And I think it's going to be really informative. And so certainly if you want to learn more as well about control strategy development, there's a couple of webinars that expound upon and continue upon some of these, what I've discussed in my presentation here today. I feel I'd be remiss if I didn't mention something related to the ongoing pandemic and COVID-19. I know I didn't gear this presentation for that purpose, but there are some recent articles on managing risk and uncertainty from some colleagues of mine at ValSource. So I've got 3 examples here that I think were really good examples of application of risk management, understanding uncertainty and working and making decisions under these stressful conditions at this time. So at this point, I just want to say thank you. My contact information is here. There's a few questions that we're going to do a little Q&A here with amongst Brian and myself, which hopefully will be helpful for you. But if you do have any other questions, please don't hesitate to contact me directly, or contact PDA as well, and we'll have some information for you so that you can do that. Thanks.
Excellent. Well, thank you very much, Darius. I greatly appreciate you going through this. This for the second time
Sure.
that we did this webinar. But in lieu of, unfortunately, we didn't get to do any live questions. We're going to be seeing what we can do about that. And thank you for offering the ability for people to ask you particular questions and reach out to you directly. In lieu of broad-based questions, there are a few questions that I figured that I would ask now that are related directly to your presentation. Maybe you could just answer those, and hopefully this is representative of what the kind of questions that you would get from our audience. So the first is that you spent considerable time discussing critical quality attributes. And of course, CQAs that are approved by regulatory authorities are product specifications. So with that, how are product specifications managed within the control strategy during your clinical development?
Okay, yeah, this is a good question. Product specifications these are tests that are associated with the quality attributes of a product. They're used in the confirmation of the quality of the product with every single lot that is produced as part of your product release. So it's important to understand though that product CQAs are not specifications. Okay, these specification tests, they're part of and are another important piece of your overall control and your overall control strategy of the product. With cell therapy products, perhaps even more so than with other types of medicinal products, due to the inherent sources of variability that can impact final product quality. For instance, like I was talking about in my presentation, the starting materials. It's really important to develop a strong understanding of those sources of variability and to frequently review your specifications that you've set for your product prior to finalizing any of your release specs, especially when you get to the point of late stage clinical and when you're going into your process qualifications and getting readying yourself for commercial purposes. Yeah, thanks. That's a good question.
Thank you very much for that. So I guess second to that one is I just want to reach on something, and you mentioned that you could get into this for a full hour afterwards, but we don't have a full hour on that. But you discussed the need. This is dealing with the vectors, the viral vectors. So you discussed the need for a raw materials control strategy, especially with regard to these vectors. So could you briefly discuss how one would apply a risk-based approach to the manufacture of one of those particular vectors that's needed in your cell and gene therapy manufacture?
Sure, yeah, absolutely. And I probably would need more than an hour to actually address this topic. We could take even a day, Brian. So I would just say, the same type of risk-based approach that I've been discussing for cell drug products, that can also be applied to the manufacture of and the development of the manufacturing process for a vector. In general, from an assessment of the criticality of the vector on cell product quality, there's a high impact to many of the product CQAs based on that vector. For example, the transduction efficiency, that certainly is one CQA of the drug product that's impacted by vector quality. For this reason, I'd say it's really important to have a sound control strategy that we can apply to the vector manufacturing as well. Probably should note here that vectors have been identified by health authorities as a starting material for the purpose of genetically modifying cells for ATMPs. Health authorities have identified these as drug substance, and thus from a regulatory perspective, the amount of data required for supporting a licensing application is similar to that of other medicinal products. So control strategy development for the purposes of vectors is also extremely important. Thanks.
Excellent. Well, thank you very much for that. Yeah, I knew that was a long, very open-ended question. So thank you for being as brief as possible for that. But yes, if anyone wants to learn more about that, I'm sure that's something that would be dealt with in more detail during the ATMP webinars in June. So finally, in the interest of time, we'll just sort of ask one more question, then we'll kind of end it all in there. In many cases, you hear all the time, the process is the product when it regards to cell and gene therapy. And the reason is because the manufacturer the final product is so incredibly dependent upon the manufacturing steps that are used to make it. So if you look at your development stage, for example, your development stage 1 is typically goes through your preclinical, your phase 1 and your phase 2 safety efficacy study. So considering that you're still making iterations in here and the process is the product, your product, how do you manage all of these? In other words, during this development stage 1, how do you manage these particular process changes? And when is the control strategy ready for life cycle stage 2 when you're locked down? If the process is the product, when is that process and product good enough? And when can you move that on to life cycle stage 2?
Sure. Yeah. This is a big challenge for cell therapies and really also for all ATMP products. So my strong suggestion here is to try to determine an initial set of CQAs as early as possible in development. Even prior to your initial clinical manufacturing, so that when process changes, and they will be coming, they will be needed. when any of those changes are proposed through your clinical manufacturing, that set of CQAs that can be used for the assessment for the potential impact and to determine how significant those proposed changes are to the product. And that will also that will help you with determining and gathering the right type of supporting data so that you can support that implementation of the change. And even potentially, based on the significance of that change, potentially use those CQAs to better understand how to design something like if a formal product comparability study is required. Additionally, identification of those initial set of CQAs and trying to understand the process as much as you can, as early as you can, it's going to help with other studies. And I showed those examples of other areas where the control strategy is defined, for instance, around contamination controls and around setting an analytical methods control strategy. The earlier you can start working on those, the better as well. As for when the control strategy may be deemed kind of quote unquote ready for stage 2, I hear a lot of people say, hey, when can we lock down the control strategy so that we can do validation, be ready for that. Using a risk-based approach for this is important as well, and really should be applied to determine your readiness of that control strategy for the purposes of stage 2 and performing a qualification activity like the process performance qualification campaign. The goal of process performance qualification, or the PPQ, is to demonstrate process consistency. And so you need to, the many sources of variability that we're dealing with these types of products, as I've been discussing throughout, that can impact product quality and process performance. These need to be well understood and addressed by your control strategy, or you would risk a possible failure to your PPQ, and that would also be critical and be something that would impact your overall project timeline, and that's critical for your overall CMC development.
Excellent. Well, thank you very much for that. So with that, I think, again, in the interest of time, we'll go ahead and end this webinar. But I wanted to thank Darius for doing this not once but twice now, this webinar. Apologies to everybody who the previous technical difficulties that we had with the webinar. Hopefully this works out well and everyone can access the link. And please, as Darius has said, if there's any further questions, you can reach out to the Pacific Northwest chapter. You can reach myself or Darius, and we can address any questions you have with that. So with that, I'd like to thank Darius again for this most informative presentation. I'd also like to give a plug for our PDA Pacific Northwest chapter. We had planned on having this as a live event, and with everything that's going on, we were unable to do so, but hopefully this information is available and is available to our members and non-members alike for this particular thing. But anyone that's in the Seattle area or the Northwest region in general that's interested in presentations such as this should please reach out to the Pacific Northwest chapter and join. And we're looking at bringing on volunteers and really expanding our chapter and providing more of these types of webinars, both locally and through more national webinars as they become available. So with that, I'd like to thank everybody for their attention, and have a good evening, and good morning, good afternoon, whatever it ends up being that you decided to watch this webinar. Thank you very much.